about some of the work that we've been doing for the last couple of years, motivated by the interaction between cancer cells and the immune system. If I were more timely, I would have something to say about the interaction of viruses in the immune system, but you know, I don't have that short a time window for getting anything useful to say. So we're going to deal with cancer and the immune system. The cancer community over the last few years has gotten very excited about the idea of using the immune system, and in fact trying to sort of modulate the immune system in order to attack cancer cells. And here I've just put a couple of pictures. A couple of years ago, Science labeled cancer immunotherapy, the idea of using the immune system to attack cancer cells, as their breakthrough of the year. And then that was followed by the Nobel Prize in Biology being awarded to Jim Allison, who was the person most responsible for reinvigorating this field over the last few years. And the idea is quite simple. The idea is that the immune system is quite powerful, and not only is it powerful, but it also can learn on the fly. And so it is sort of a match for cancer. If you try to treat cancer with simple small molecule drugs, what the experience has been is that those drugs work for a while, a year, 2 years, improves the life of the patient, but eventually the cancer cells can find ways around the action of those drugs, either by genetic mutations or by phenotypic changes, and so the drugs stop working at some point. The immune system is smart enough to respond to changes in the cancer by changing its own strategies and hopefully keep up with cancer. Now, when you think about the cancer interacting with the immune system, you have to imagine that this is actually a very multifaceted problem. The problem ranges all the way from the molecular level, which is what I'll talk about today, where in order to, for example, detect the cancer cell, you have to bind the cancer cell with an adaptive immune cell, typically a T cell, which is what we're talking about here. But this is only the lowest level. At the level of cell biology, there are direct pathways which determine if the activated T cell will, in fact, kill the tumor cell, and that's a very complicated story. And it was this part of the process that actually was the contribution of Jim Allison in understanding these different pathways and in coming up with drugs that could unleash the immune system that otherwise would be inhibited by these pathways. But it doesn't stop at the level of cell biology. At the level of tissue biology, or even, you can even think of it as ecology. If you think of the neighborhood of a growing tumor, there are all sorts of different types of immune cells that interact with each other and collectively decide how much of a threat the cancer is, what it's going to do to attack it. And here are some labels of some of those cells: macrophages, neutrophils, which are part of the innate immune system. Then there are natural killer cells, dendritic cells. So at the higher levels, it actually becomes a collective decision made by a arrangement of cells, not just each individual cell deciding what to do. And of course, the goal is, sort of looked at in the sort of bottom right panel, is what you really want is you want the immune system to win a battle that it should be able to recognize and kill the tumor cells, as opposed to the other battle, which is that some subtype of the cancer cells can evade immune detection, eventually take over the population, and grow out of control with no effect of the immune system. So at the molecular level, the most fundamental question is, how do you detect a tumor? And the reason that's a fundamental question is that tumors don't look at the cellular level very different than our own cells. There are a few mutations that change various signaling factors, but on the whole, tumor cells are similar to our own cells. So what is the leverage that the immune system has for detecting a cell as being part of a growing cancer, and in particular then deciding to kill it? So the way that T cells detect other cells, we'll see this in more detail in a few moments, the way that T cells detect other cells is cells have a general machinery that displays on their surface various pieces of proteins that are being made within the cell. I liken it to many, many years ago. I think the first time I visited Japan, I was amazed to see that trucks, when they drove on the highway of Japan, had a neon display on the front of the truck displaying what speed they were going at in order to, make the jobs of the police officers enforcing the speed limit easier. So cells do something similar. They take information about what's going on in their interior and display it on the surface for the immune system to detect. Now, if those pieces of proteins on the surface are normal pieces that the immune system expects to see, being the protein pieces of our normal cells, then of course the T cell is supposed to not attack. On the other hand, if it sees something called a neo-antigen, which is the modified protein, because the tumor cell has mutated that peptide, then in fact it's supposed to kill that. So tumors can be detected by changes in the peptide sequences of things that are displayed on the surface of cells in your body. Now, the fact that this works has been known for a long time for various indirect reasons. For example, tumor immunotherapy, this sort of advance in how to get the immune system to attack tumors, success correlates with how many mutations the tumor cells have. So for example, melanoma, which are cells in your skin that are exposed to UV radiation that have a huge number of mutations, those are the easiest cells for the immune system to detect, and that is the type of cancer where immunotherapy has been the most successful. In cases where the immune burden, the mutational burden is lower, where the number of mutations is smaller, and therefore the cells look more closely like normal tissue cells in your body, then in fact immunotherapy has been less successful. And so a couple of other pieces of evidence, you can of course directly look for T cells in mice that detect cancer cells via their being neo-antigens, via these modified peptides. From one particular paper just in 2014, I don't want to go through the details because I don't have time, but they just explicitly found specific T cells that did recognize tumor neo-antigens, sort of a direct measurement that there were T cells that were able to specifically recognize cells in a particular mouse model of cancer. More recently, my group has looked at something which is again an indirect way of sort of showing this interaction of the immune system with cancer. What you see on the top here are pictures of tumors taken from actual patients. The blue is the tumor and the yellow are the T cells. And what you see going from left to right is an increasingly successful meeting up of the T cells with the tumor. On the left, the T cells are somehow being excluded from the tumor volume. In the middle, the T cells are sort of partially getting in, but are mostly relegated to these black outside regions on the tumor. And then all the way to the right, then you actually have situations where they're fully able to encounter the T cells, and you can analyze these situations. But the bottom line is that patients that had situation number 3, where the T cells are fully able to encounter the cancer cells, those are the ones that have the best prognosis. So this is data taken a long time ago, and we know the outcomes of all these patients, and good prognosis always correlates with having the most engaged immune system in actually being able to interrogate the cancer cells. So let me talk about how this works at the molecular level, because the question then is, well, how do you detect tumor cells? And I've just shown you some evidence that you do detect tumor cells and how at the same time do you avoid killing your own cells? So this is called, how do you distinguish self from non-self? And so the player here is a T cell, and here is on the left-hand side of the slide is a diagram at the molecular level of what that recognition event looks like. So on top you see a sort of large-scale image of a T cell interacting with a peptide, and then you see a blow-up picture on the bottom. So what you see on the bottom is you see this sort of linearly splayed out peptide, usually 10 or so residues long. It's placed in the groove of a large molecule called an MHC complex, and it's displayed for interrogation by these loops of the T cell receptor. So there's a CDR3 loop, the alpha loop, and the CDR3 beta loop. These loops are different in every single T cell in your body, and this serves as a sort of interrogator of this peptide. And if there's a strong binding event between the T cell and this peptide, then in your peripheral body and in your tissues, then the T cell is activated and told to kill this cell. So how do you avoid killing cells that have a T cell that's able to detect a peptide that's supposed to be there? Right? This is the mechanism that's supposed to attack and kill cells that have peptides that are not supposed to be there. How do you prevent it from killing cells that have peptides that are supposed to be there? So the answer is the T cells go through a training exercise in the thymus, which is why they're called T cells. And the training exercise is called negative selection. And what you do is you take the peptides in your body that are most commonly displayed, your normal proteins, you put them out on training cells. The T cells go through the thymus, and if they're strongly detecting one of those cells, then those T cells are basically told to commit suicide. So any T cell that actively detects a self cell is deleted out from the list of T cells, from the repertoire of T cells. And all the T cells that don't do that, the T cells that are functional but are not strongly detecting something which is supposed to be there, are then released into your body. They go hunting, and if they can find a thing that's not supposed to be there by binding strongly to it, and they already know that they don't bind strongly to something which is supposed to be there, then they're instructed to go kill the cell. So that's in a simplified version of how the molecular level detection works. And this does work. So we want to try to understand how this could work on the molecular level. What is the mechanism by which you could have a selection procedure which would eliminate most of the self peptides from being detected, but would nonetheless allow you to detect tumor neoantigens? And just a reminder that tumor neoantigens are mutated peptides. Typically, the mutated peptide is differing from the non-mutated peptide by one residue. So it's a detection problem where a 10 residue amino acid chain is not detected, and you change one of those residues, and on average, now you are detected by the system. So how does that work? So here is a schematic, here are two schematic pictures of how to think about the problem. And the question is, how does negative selection at the end of the day work? And I want to use a somewhat gory example of these two schematic ideas. The gory example is you have this crazed person who wants to attend, some major sporting event, in the old days when those things existed, and wants to kill all the physicists at that event. Because, whatever reason, he just had a very bad experience in physics in high school. So how is he going to know in this stadium of 50,000 people, how is he going to know who are the physicists? So there's two strategies you can imagine. Strategy number one is he can visit all the physics departments in the city in question. Let's say Boston. He has to visit 5 or 6 major physics departments in the city of Boston. And he'll sit there outside the physics department office, and he'll memorize who all the physicists are going into the office. He'll just look at them. He has a good memory. He'll just remember them, and he'll search for those same people at the stadium when he goes to there. So that strategy is a good strategy, making sure that you don't kill other people. Because if they don't happen to be in the physics department, in the physics office as a faculty member, then you're not going to kill them. On the other hand, people are in trips, they go away, not everybody shows up in the department office, so you might miss a large number of physicists if you do that. So that's a memorization strategy. A different strategy would be that you spend a lot of time studying the phenotypes of physicists. You learn what physicists look like, how they act, how they drink their coffee, how they talk to other people, and you try to form an idealized version of what characteristics would enable you to detect physicists in general, without memorizing any specific ones. And now you go to the baseball stadium and you look for people who have all those characteristics. Now here you have the opposite type of error, because now, you probably will get most people who are physicists, assuming that such characteristics exist. But lots of other people might also fall into that category. You might have lots of engineers, lots of chemists that sort of look similar enough to physicists that you're going to get rid of them also. So in our conceptual framework, in that conceptual framework, you can draw a diagram of how your recognition is going to work if you are vary away from something which is your natural state. So in the memorization rule, if you change one peptide away from your baseline thing that you are recognizing, changing one peptide will mean that you don't recognize it anymore. So that's good for detecting tumor neo-antigens. You've deleted everything that memorized the list of peptides that are self-peptides, and now you change one of that, you no longer have that in your list, and now you detect it and you can kill that cancer cell. The other possibility is where you have this more broad detection scheme, and now your detection scheme is probably not going to be good enough such that changing one peptide is going to give you such a different set of properties that you'll be able to tell the difference. So our hypothesis, based on also some simple models, which are in the sort of random energy model arena, our hypothesis would be that this would be a more likely scenario to be able to detect tumor neoantigens. And one consequence of that is what I said before, is you make errors that because you can't see every single peptide, that you actually don't completely get rid of all the T cells that detect self-peptides. You get rid of most of the most common self-peptide detecting T cells, but there always will be errors, and that's actually known. So if you look at the immunology literature, and a paper written some number of years ago called Not So Negative Selection, they show that this process has the type of errors that you would expect for that second conceptual type of detection, the memorization detection. Now, just the last 5 minutes, what we've been trying to do is say, okay, we have this conceptual picture, we have an idea of how it might work. Can we use molecular approaches, molecular modeling approaches to see if things really look that way? So what we did is we took data established some number of years ago of taking a single peptide listed on top and measuring in a variety of experimental ways that, again, I won't go through, what T cells can detect those peptides, and training a essentially molecular biophysics code to be able to detect ones based on this data set, and then seeing how we do more generally. So we need a molecular biophysics model. There are lots of choices out there in the literature. We took Awesome, not because it's the first one alphabetically in this list of papers, but because it was developed by Peter Wolonis and Greg Papoyan, who know quite well and were willing to help us, understand how to work this code and use it for this purpose. So the logic was we would train the system using experimental data. We would collect good binders and bad binders. We would train the parameters in this Awesome model based on that data. Then we would try to see to what extent that would work in explaining other data that were not part of our test set. And here is a results of our work. So the thing you can look across is the way these training things work and this machine learning world works is you first have to make sure that you have the ability to tell the difference between good and bad binders with your energy. So that's what the first graphs on the left are. For 3 different peptides, you see that you can get a trained model that gives you a pretty large distinction between good and bad binders. And then what you want to do is you want to take data that's not part of your training set and see if it still works. And again, skipping the details, we can show that this works reasonably well and can work better to the extent that you know more details about the crystal structure. Now, just one more point is that model gives us the ability to then try to understand the system on this larger scale, on the scale of the entire T cell repertory, which is very large. In fact, if you look at the numbers, the size of the repertory typically are 10 to the 8th, 10 to the 9th different clones. You do negative selection with 10 to the 4th different peptides. You can't do this if you're going to do all atom simulations and run a serious molecular biophysics code. What you need is you need essentially something can just be evaluated very simply, some type of simplified model that then gives you the ability to have all those different pairs calculated. And the answer to the question of, are you close to schematic A or schematic B, the easiest way to get at that question is to say, how good are you at detecting totally foreign peptides, as like might come, for example, from a virus, compared to detecting a tumor neoantigen, which is differed only by one. And what you see in these graphs on the right in particular is that over a wide range of numbers and calculations, whatever, it's only a factor of 2 better to detect foreign versus mutants. I think we have the graph upside down here. But it's not really a big difference. So the system really is behaving, at least in this model, via this memorization paradigm. And this, of course, is still a hypothesis. There is other supporting evidence for this hypothesis that one can see whether it's even possible to find a phenotype of self-peptides that somehow makes sense. So far, evidence is against that, but at least this gives us a way to begin to think about this question. So those of you interested in this subject, and this was a sort of short presentation, trying to keep more or less, I think, to 20 minutes. If those of you interested in this question, we've written a number of papers at different aspects of this paper. The first one is about this simple conceptual model. Then the data I showed you on actual T cells interacting with human tissue samples, human tumor samples, is sort of the second paper. And the most recent paper is a preprint done together with Jose and other people, is this attempt to use this molecular biophysics model to see to what extent this conceptual picture might be right. And lots of people worked on this in various ways. The most recent work on this molecular biophysics model has really been done primarily by Jing Chen with the help of Jason. Jing Chen was a student of Jose and now is a postdoc in MIT, continuing to work on molecular biophysics things. So with that, thank you very much for your attention. I hope this was understandable, and I'll be happy to take any questions either in person or by email afterwards. Thank you. Anybody still there?
Other questions? It was so brilliantly clear.
Well, yes. So if anybody has any questions that want to send me by email, that would be fun.
Can I ask a question?
Yes.
It's Bob Austin speaking.
Oh, hi, Bob.
Hi. I think it's sort of becoming clear, and it's a really nice talk. I enjoyed, but I think eventually these immune therapies seem to be failing. Is that right? Or what do you think the...
I think the story is mixed. I think that the hope that this would be a panacea for all cancer types, I think is no longer, I think there are lots of cancer types where it just doesn't work at all for reasons that are not completely clear. So, you know, the cases that work and the ones that you hear about, you know, on the TV ads for immunotherapy drugs are melanoma and various types of lung cancer. But in some other very popular cancers, breast cancer, pancreatic cancer, gliomas, they don't seem to work, and nobody really knows why. So I think the sort of home run, magic bullet idea is probably not going to be true, at least immediately. And then even those other cases, even in immunotherapy, there's still a large number of patients, 50%, 60%, that don't respond as well. So the great breakthrough was having small numbers of 30%, 40% respond, but respond in a way that looked like it was going to be a pretty permanent response. Unlike drugs where after a year or two, you would go back to being immune to the drugs, and it looked like you were getting long-term response in many of those cases. I think that's still true in some cases. So it's a sort of mixed story. It's not the magic bullet that's going to kill all cancers of all types, but there still is evidence that when this works, it can really provide much longer term benefit than a typical drug therapy. So we'll see. I mean, I think there's still a lot of work going on in all sorts of directions in the cancer community. As usual, whenever there's a sort of partial success, then a lot of the work being done is very empirical. Just try all sorts of combinations and things and see if you can improve the empirical results. That will die down after a while in favor of perhaps trying to understand things a little bit better from a more mechanistic perspective. So I think we're sort of near that transition point these days.
So, Herbie,
Can I ask another question?
Jose,
so just a fact,
can I
go ahead.
ask him? Jose.
Go ahead. Go ahead.
Yeah,
Okay. It's I don't know what you're drinking there, Jose, but
it's breast
I'm just
milk.
thinking.
I'm going to ask you a very naive question because I don't really know the cell biology well enough. Are the presentations of the cells, either cancer or normal cells, changing dynamically throughout the cell cycle? And if so, how does if that is true, how does the T cell?
I don't think so. I think cells tend to, I mean, I actually don't know for sure, but I don't think the displays of what peptides are on their surface change over the cell cycle. They might change, I mean, they certainly change when cells differentiate from one type to another. So it's not like they're permanently, it's not like you have a stem cell and you permanently decide what to put on the surface and then it differentiates and stays there. So there's some dynamic process of taking the receptors, whatever, endocytosing receptors and putting new ones on the surface. But I don't think it's on the time scale of the cell cycle. I think it's a much longer time scale having to do with cell differentiation and that sort of longer time scales of weeks or longer.
And the concentration of the various things that are being presented, is that basically the same in both cancer cells and regular cells?
There are some cancer cells that have a strategy of down-regulating some of that presentation machinery. So that can happen. But typically, at least from what we've been able to determine, when cancer cells are in their growing, rapidly growing primary tumor mode, that doesn't tend to happen. They tend to be about the same as the original cells that they came from. Where they seem to be able to downregulate, and this presentation machinery is at later stages when cells begin to metastasize. There's some evidence that cancer cells can fight back and try to prevent.
Let's try to squeeze one fast question by Ilya now before he changes the topic.
Yeah, good. Yes, Ilya.
Ilya.
Yes, hi. Thank you for a great talk. So maybe a naive question, right? So in this discussion that you are presenting in this mechanism, what prevents the immune cells attacking cells inside the body that are, let's say, aged cells, right? Cells that accumulate non-cancerous mutations that, eventually many cells do.
Nothing in principle. I mean, I think the question is how many.
Happen?
Does it happen? I'm sure it does. I don't know the details for sure, but I'm sure it does. On the other hand, I mean, exactly how, I mean, melanoma cells have a huge enough, and also, non-small cell lung cancer, which are the two primary ones where things work the best, have a very, very large number of mutations. I don't quite know, I mean, I should check how many mutations a typical cell picks up as a function of age. I don't know if it's comparable to some of these extremely mutagenic tumor. I mean, cigarette smoke and UV radiation and those things really create a very, very large number of mutations, such that even if you take, 10 peptide sequences, you actually, relatively often get changes in amino acid. I don't know the numbers for aging cells. It's a good question.
Okay.
Thank you, everybody. And certainly for any students, if you're interested in this topic, just send me an email and I'll try to respond.